Good afternoon, everyone, and thank you for joining. My name is Kayla Valdez, and I am the xRNA Program Analyst here at NCATS, and I welcome you to today's presentation. Just to tell you a little bit more about the ERCC, it stands for the Extracellular RNA Communications Consortium, which is an NIH Common Fund program which works to advance science and research of extracellular RNA. The consortium hosts monthly presentations on a variety of research topics, and I'm pleased to introduce today's speaker, Jack Wiedrick from Oregon Health and Science University. The title of his presentation is Robust Biomarker Selection from Real-Time qPCR Data Using Statistical Consensus Criteria. Jack?
Hi, thanks, Kayla. Thanks to everyone for attending. I am a statistician at the Biostatistics and Design Program at Oregon Health and Science University, and this is kind of an extension of a talk that Jody is the director of our program. She and I developed this stuff together. We presented a sort of a shorter version of this in a kind of a truncated format, I guess, earlier this year in February. This is a much extended version that also includes some kind of new methodology. As Kayla said, it's basically dealing with biomarker selection, and the problem here is where we've got sort of maybe a semi-large dimensional kind of space. Like we have all kinds of candidates, potential biomarkers. We're not sure if there are any biomarkers among those candidates, or if so, which ones they are. And it's too expensive or time-consuming or whatever to search through all of them. So you have to do some kind of selection, and this methodology is about that. How do you do selection in a sort of fair way and a robust way? I'm going to talk a little bit about what that means. Hopefully the goal is to minimize false positives. You don't want to chase down rabbit holes. You want to quickly hone in on the things that are likely to be real biomarkers and exclude things that are not. One thing, just as a quick comment before I start, something that will not really be presented. You won't see anything about p-values or false discovery rate corrections, and that's very deliberate. It's because Jody and I are kind of one of I think a growing number of statisticians who really don't believe in that stuff very much in the selection problem. We think that selection is sort of a more difficult problem than that, and that simple methods can't always be applied. So what we've designed is kind of a methodology that I think has been developed over the last 3 years, at least formally, and we're still tweaking it and working on it, and so far it seems to be doing quite well. So we're kind of happy to present it. We'd like to sort of get it out there. We think it's a good way of going about the selection problem. So anyway, let's go ahead and get started. Quick background, we're looking at microRNAs. Sorry, let's go back in this case, but it could be, this kind of methodology could be applied to any problem, really. Essentially, we'll get expression values or some kind of other continuous type value that essentially is a set of numbers measured on patients, subject samples, whatever. And we want to choose the ones that sort of separate groups, say from, healthy from diseased, or, a case from a control in more general terms. In this specific case, the methodology has been, like I said, detailing the specific problems, I guess, associated with RT-qPCR data. But, like I said, some of the same considerations apply to any kind of data. Another thing that's going to come up is that our particular project was divided into UH2 and UH3 phases. The UH2 is a discovery phase where we're sort of searching for potential biomarkers, and the UH3 phase is the one we're currently in right now, where we're sort of validating our choices in independent data. So the whole point, especially the problem mainly, like I said, that this talk will be dealing with is kind of what happens in the UH2 phase. And the keywords that I want you to hone in on are the words robust and select. What do we mean by robust? A robust marker is one that's not sensitive to irrelevant stuff in the samples. By irrelevant stuff, I mean that we usually have to be selecting these things from relatively small samples, say like 100 patients or 200 patients. May or may not be representative of the eventual population we want to apply these markers to. And sometimes those small, in fact, usually those small samples will have lots of irrelevant features, just sort of random selection issues, or sorry, sampling issues that highlight certain things that are not relevant to the markers, but that sometimes the markers will hone in on. So we want to find markers that don't care about stuff like that, and instead hone in on the signal that we're really interested in. That is the group separation signal. The whole problem is this, like I said, it's selection, the fact that we have to select. If we could just sort of measure everything and understand how all the processes work in the disease, or how the markers were relevant to the disease, and specify a clear model, we wouldn't need to worry about this stuff. But we never know any of that in advance. We always have to make a guess that there's going to be some kind of like biomarker in this pile of candidates, and use some incomplete information to make guesses about, like I said, to zoom in on the ones that we like and dismiss the ones we don't. The problem is that in these small samples, these irrelevant features that I mentioned before are often really prominent, and so that's kind of the reason why we have a problem with reproducibility in a lot of science, because so much of it is done on small samples, and so many of the findings are really just spurious associations that are, small sample features. Anyway, and then a little bit toward the end of the talk, we'll also talk about sort of validation, things to look for in validation that are going to be important. So our particular scenario, and the one that motivated the development of most of this methodology, is that we're looking for markers of Alzheimer's disease. And in this case, the biofluid we're interested in is cerebrospinal fluid, although we're now trying to expand that into serum. And we just want to know, like, are there any microRNA markers, or any sort of, can microRNA expression in the cerebrospinal fluid distinguish Alzheimer's disease patients from healthy controls? If there are any markers like that, how do we find out? I mean, how do we discover them? We started out by just sort of getting an off-the-shelf array that included a large panel of microRNAs, and measured them all in all the groups, on all the samples, and looked for some group differences. The problem, again, in this, focusing on the left half here, the phase 1 discovery, is that we had a large set of them. There were, like I said, about 750. We don't want to chase down every single one of those. We don't have time and resources for that. So we want to quickly sort of filter through them and see which ones look promising, and sort of see how promising. And then after we've gotten a smaller set of ones that we think maybe are relevant or promising, then we sort of do a verification or validation on those and see if our initial sort of assessments were correct, or at least in the ballpark. In our particular case, the panel that we used sort of had, a large set of probes and a number of cards, but because there are so many probes, we can only fit sort of one sample per card. So we only had one measured sample per probe, per sort of, subject sample. Some of those probes didn't yield any value, so we have missing data problems, and no explanation for it, right? Is it because there was no expression? Is it because the expression was just too low to be measured? Were there technical assay failure problems? And those happen as well. Also, some of the values that we do receive are untrustworthy in one way or another. Quality metrics from the device that measures these things give us some indications that, okay, maybe this wasn't well measured, or something may have happened. There may have been contamination. There are a wide range of things that can go wrong with experiments like this, and some of those things will affect the values that we receive back in the data. Also, sort of the biggest problem that relates to our main thing is that most of the things that we're measuring are probably irrelevant. We've got 750 things. We don't really want 750 things. We'd rather have close to a dozen, maybe even less. So we want to quickly throw out as much as we can and focus in on the stuff that we like. And as I mentioned before, this doesn't only apply, what I'm going to describe doesn't only apply to RT-qPCR data. You can, some of these considerations may be different for a different data type, but the selection methodology is, I think, more generally applicable. And, okay, so what is it then? We want robust selection. So what do we mean? We want to emphasize predictors that we care about and quickly throw away the ones that we don't care about. We also want to have, and here's where the statistics comes into play, we want to understand our uncertainty about the prediction model. For example, if we use these markers to form a prediction model for the disease or, a feature of interest, we want to know how confident we can be in that. And the confidence kind of comes in two places. There's one, like how confident that a particular marker is giving some signal to distinguish the groups, but also sort of how confident we can be that the particular markers that we think are giving that signal are even relevant or operate jointly or something like that. There's a nested kind of character to that uncertainty as well. Another thing that we want our statistical models to do, rather than just select, we also want them to provide some predictions. That is, predictions for how well these markers will perform in an independent data set. And we want those predictions to be testable and realistic. I mean, they shouldn't be so unwieldy that we need massive data sets in order to verify them. So again, what our pipeline does is produce that and give us very sort of specific predictions that you'll see in the validation portion of this talk become really helpful. And again, going back to the talk, how do we do all this? We just call it a consensus methodology, which means that we use a lot of different kinds of statistical tests and sort of combine the results. In other words, getting a consensus opinion from various different judges, I guess. You can think of each of the tests as sort of a judge or a panel that rates each of the markers on how well they do. But each judge is a little bit biased. So in order to get a better, more unbiased opinion, we can sort of ask all of the judges and sort of take an average of them in some sense. Just as a little bit more background, why aren't we just using t-tests or something like that? Those are kinds of standard methods that are used in these kinds of problems. They've been used for years, but the reason we don't use them is because we don't think they give good answers. It's not that the tests themselves are flawed. They do give good answers to different kinds of questions. But the answers they give are the wrong questions in this case. We're asking for kind of a model selection question, and a t-test or a null hypothesis test is not appropriate for a model selection problem. And here's a little bit why. For one thing, a p-value just says something about statistical significance. It doesn't mean it's predictive at all. Here's kind of a quick example where you've got a distribution of values and controls and distribution in cases. Their centers, their means are very, very different, extremely statistically significant, but it's completely non-predictive. If we just took a single measurement, we would have no way of knowing whether that measurement was a case or control. Also, as I mentioned before, our values aren't completely trustworthy. The values we get back from the measurements are not all correct, whereas the t-test, however, assumes that they are. And it turns out that's a very big assumption. One way in which, for RT-qPCR data, one way in which they're not correct is that we have a censoring problem. That means as we're amplifying the sample through different, cycle upon cycle upon cycle, eventually we start just amplifying noise. We can't continue that forever. So we have to stop after a certain number of cycles, and if we don't see any expression beyond that number, we can't necessarily conclude that there's no expression. All we can conclude is that it's lower than that. So at some point, like I said, the noise will dominate the signal, and we need to sort of stop our testing at that point, but that doesn't mean there's no signal. It just means we don't know how strong, or in this case, how weak the signal is. So in other words, the values we would get back from these measurements, which in some way the cycle time is kind of a proxy for expression, and that high cycle times mean low expression. In the case of very, very high cycle times, we don't necessarily know what the expression is. We don't have any way to map that in a direct way. We just know that it's got to be smaller than what sort of the largest cycle time we attempted would imply. Another problem with these kind of like screening or filtering using t-tests and p-values is that it's kind of a one-at-a-time methodology where you just sort of test each marker, but that can miss really, really large parts of what's going on. Here's kind of an example where we've got an outcome, let's say. In this case, you could think of the outcome as like being the severity of a disease or something like that. And we've got one microRNA, microRNA1. We've plotted the outcome values against the values of the microRNA1, and it looks like there's no association at all. There's completely like just a random scatter and doesn't seem to be any trend. But the problem in this particular case is that there's another, say, marker, like let's say microRNA2, that's associated with both the outcome and, in this case, this other predictor. If we knew the values of both markers, we would be able to make an actual prediction. In this case, if for any particular value of the second hidden marker, the one that is not included in that test, say, for example, if we condition on that, say like, oh, we're only going to look at the high values or look at the low values, suddenly microRNA1 becomes very predictive. There's now a strong trend with the outcome that wasn't visible in the aggregated data. That's an example of something that's called Simpson's paradox, but really what it represents is some kind of like a hidden confounding that a one-at-a-time test ignores, doesn't measure, and so it can lead to wrong conclusions. So we also want tests that are going to look at markers not only in isolation one at a time, but in combination, and sometimes in complex combinations. So, okay, here's our methodology, sort of in a nutshell, at least the steps. What do we do? Well, like I said, Jody and I are both of the mind that just doing a bunch of null hypothesis tests is not the right way to go about things. We prefer visualizations, actually looking at the data, trying to understand what's going on in the data. And a lot of the tests that I'll be sort of presenting really boil down to that. If you were able to sort of plot whatever that test is measuring in some way and look for strange features like blobs or trends or whatever, that's sort of the result that the test is giving you back. And so we emphasize visualization quite a bit, and partly in order to understand sort of what's happening in the data, maybe like I said, looking for signals, but also to look for batch artifacts, which can be large in this case. So as I mentioned before, in this particular case, we had 2 cards per subject. Each card represented a different sort of batch, even though it was the same subject sample on both cards. And those 2 cards would have to be, run at slightly different times. We had a large number of samples, so they had to be run over many different days where the machine is going in and out of calibration, et cetera, et cetera. All those things we should look for in the data and try to model them out if we can. That's sort of step number 2. We try to normalize the data in some way by understanding sort of the batch influences and also looking for other kind of sources of technical noise. We want to do that before we start any selection because the values before that are kind of biased by these batch of artifacts, and we want to get rid of that before we even start to select. The third step, which I think is a very important one as well, is to immediately filter as much as you can. So if you notice some data problems, for example, things that don't seem to assay well, for example, all of the samples seem to have very low expression, or there's a lot of dropouts, missing values, or whatever. If you're going to try and use this technology as a diagnostic tool, then those kinds of things can't really be biomarkers because you can't trust that it would work on any patient anyway. So you may as well just get rid of them from the start. So reduce your problem by eliminating these unsuitable ones. The fourth one is kind of the one that's the main point of the talk, is use multiple statistical methods to make selections. The reason why that's robust is because if something is showing up in many different methods, regardless of how you look at it or what angle you look at it, then the signal is probably real. It's probably not honing in on spurious things that the assumptions of the methods are causing it to focus on, if that makes sense. Each method gives a slightly different perspective, and by combining all those perspectives, you can get a clearer overall view. The fifth step, I think, is also very important, and it's one that a lot of methodologies overlook, in that you want to have, again, testable predictions in your discovery phase. So after you've selected a set of markers, you want to do some kind of what we call cross-validation. That means using the existing sample to try and, basically, it's kind of like a bootstrapping type technology where you resample randomly and try and use parts of the sample to predict the values of other parts of the sample. By doing this over and over, you can get kind of a feel for how well the markers are likely to do in an independent sample. Like I said, that gives you a testable prediction. You also try and rank or prioritize the markers that you found in the discovery phase and see if those, if that prioritization holds up in validation. And the last step, of course, the UH3 phase is validating, where we actually test all those predictions that we had in the discovery phase and see if we find other unusual things. This is also the point at which we look for sort of covariate influences. The reason why we're not by covariate things like what if the markers behave differently for this, males or females, or only work well at different ages, or other kinds of biological effects that are happening that are maybe not relevant to the disease but might be relevant for the markers. The reason why we're not looking for all that in the discovery phase is, again, because we've got such a large set of markers, we're probably going to find stuff like that, and it's probably going to be spurious. So we want to sort of simplify our problem in discovery and only kind of worry about those nuances and complexities later on, once we've got a set that we trust a little bit more. So steps 1 through 5 are, like I said, the discovery part, the UH2 part, and step 6 is the validation part. So let's just go kind of in order, all these steps, and I'll sort of demonstrate a little bit about what I mean. The point of visualization, like I said, especially early on in discovery, is to just find heterogeneities or failures. Look for things that are wrong in the data and get rid of candidates that don't seem to be doing well, that have problems. We could spend a lot of time and money chasing down those problems, but ultimately we don't want to. We'd rather look for candidates that are going to be robust to those kinds of issues. And so things that don't seem robust, things that are very sensitive to assay problems are ones that we can immediately exclude. For example, in our initial discovery set, we had about 750 candidates, and we excluded almost half of them just based on their kind of assay quality characteristics. So that was a huge reduction in time and effort. We also want to sort of look for like detection limits or thresholds, dynamic ranges, I guess, of what the machine. In this case, we have cycle times are our raw values, and we need to think about a censoring threshold. That means a value beyond which the cycle times are not accurate. The cycle times simply reflect that the expression wasn't measured. And so we discovered that by plotting our data very carefully and mapping it according to, like I said, the quality characteristics that came from the machine. We also, if we have some kind of control probes that are on each of these plates and so forth, we want those kinds of things to be uniform, and if they're not uniform, they indicate kind of batch artifacts or technical noise. So the things we kind of sometimes will influence that, and the things that we look for problems across are things like processing batches. Like, for example, if we changed reagents, a new lot of cards, for example, or a measurement batch, which would be like a particular plate or a particular day when we're running several plates. Different settings of the instrument. If the instrument had to be recalibrated, for example, that changes the settings and would be expected to introduce a batch effect. So when we find those kinds of things, like on the left panel, we model them out. So you notice that the plates and cards are sort of going up and down across some different replicates here on the left. After we put them into a statistical model, we can kind of smooth them out a little bit. Notice that not only do we line up the middles a lot better, but we also reduce the variance in some sense, and that the center parts of the distributions are tighter now. Another way that we'd like to make life easier on ourselves is to use kind of robust summaries of the data. So in our particular case, in our discovery example, we only had a single probe per plate, so there's no other summary other than what that value was. But sometimes you have things measured in duplicate or triplicate or whatever. And in those cases, we like to use what we call a robust center, a median. It's common to use a mean, but if one, let's say if something was measured in triplicate and one of those dropped out, or one of those had a very extreme value because of a problem, the mean would be hugely affected, whereas the median would not be. If the three values were all very similar, then the mean and the median should be about the same. So in all cases, the median is sort of a better choice for that kind of a summary. Also, the cycle time values are kind of awkward to work with because, again, we have the censoring problem where these high values we're not able to trust. They're also on an inverse scale, so higher CT values mean less expression. So we'd rather turn it around and have something that we would call an expression scale. And one of the features of this expression scale is that if once we've decided on a censoring threshold, that means a value like a CT value beyond which we're not going to trust the results, we, on the expression scale, we'll just set all those to 0, just say they're unmeasured expression. But those zeros don't mean they had 0 expression. It just means we don't know the expression. It's so low that we weren't able to measure it on the technology. So this is a much more convenient scale to work in and allows kind of better modeling. So here's kind of a plot of what happened. So if you notice that the low CT values are mapping, so on the vertical axis, low CT values are mapping to high expression values and sort of vice versa. And then notice that clump in the upper left corner, those are all the censored values. Those are the ones that turn into zeros. Those zeros, notice that they're across a very large range of CT values, say from like 33 or 34 up to say 45. That, like I said, having those actual values is awkward on the CT scale because none of those are really trustworthy. So turning them all into kind of a symbolic 0 is easier and makes a lot more sense for our models. Next, like I said, once we've sort of transformed and kind of modeled out all the batch effects and we've got a set of expression values that we believe are reliable, now we want to see kind of how much, I guess, how much promise each marker might have. And one of the things that you need to have for a good biomarker is it needs to be measurable in both groups. So that's what we mean by reasonably attested. If, for example, if you have quite a lot of censoring, for example, it's hard to measure this marker in 75% of the samples, regardless of whether they're a case or control, that can never be a biomarker because you could never have high specificity with a marker like that. You've got to throw things like that. Even if they've got great signal within the ones that are measurable, it's not generally applicable. It's not good as a screening tool. Also, the accuracy of our cycle times obviously should be pretty high, so we filter out the ones that are not. The censoring should also be unrelated to the accuracy of the cycle times. By that, I mean that the probability of being measured or having low expression should be unrelated to how accurate expression is quantified when it is expressed. And if we see any kind of correlation here, then that means that we can't trust the censoring. In other words, we can't know whether something is expressed or not expressed with very much clarity. So in our case, we don't see any correlation. That's a good thing. So it means that with the markers that we've got, we can kind of move forward. So after, like I said, we filtered almost half earlier on just based on sort of technical issues, and we were left with, say, 411 what we called well-measured targets. We were able to get 2/3 of those out right away on more theoretical grounds. They were unlikely to be viable biomarkers, at least using this technology. So now we've got a much, much smaller set to work on, and the problem's a lot more tractable. Finally, when we get to the actual the methods for selection, like I mentioned before, we'd like to use a lot of different tests because they offer different views of the classification. And we can have tests that kind of differ on, look at different aspects, I guess, of what classification means. one kind of question we have is individual markers, like which of the individual markers are like singly promising. And some simple way to look at that is, are the cycle time counts equal? You know, just our controls, do they tend to have lower cycle times than the cases, for example? That indicates kind of an association between the expression values and the disease. Notice that it's important here to use a test that accounts for the censoring. So what the test that we use, a log rank test, does properly account for censored values. Another thing we use is receiver operating curve analysis, where what this is basically looking for is the entire distribution. We have a distribution of values for cases, a distribution of values for controls. We want to know how separate those distributions are, and a ROC curve quantifies that, if there's separation in expression values. It looks at the entire distribution. Other kinds of questions we have are and these are the ones that relate to, like I said, the confounding and interaction and kind of a moderation type examples that I showed before, is what if we have groups of markers together? Are they highly correlated? Do they, like I said, do they condition one another in some sense? Do we get more information by combining markers than we do by looking at individual ones? And almost always the answer is yes. The ways to kind of look at that are, a very nice way to look at it is what's called a random forest, which is, I'll explain in kind of technical detail later. You may have seen random forest, but it's essentially generating classification trees, a lot of different kinds of classification trees, testing out ways of making decisions, in other words, and then averaging across those tests. They can capture, the advantage of a random forest is it can capture really, really complex cross-marker interactions that simple linear models can't. Another thing that we like to do is not just one kind of random forest, but many kinds, so alternative random forest. So what a random forest technically is, like I said, you generate a decision tree many different times by randomly sampling from the data and also randomly sampling from the candidate markers. And you average across all of those, and what that average view gives is sort of a ranking of your markers. How important are your markers for distinguishing the groups? We can have different kinds of random forests, though, and the differences relate to how the individual decision trees are constructed. So here are 4 examples that we use. CART trees, boosted classification trees, et cetera. You can look up those if you're interested. But essentially, they're the same kind of thing. They just do it in a slightly different way. The component trees are deciding how to classify slightly differently. Anyway, after applying these and sort of forming a consensus across those different methods, we see really, really great separation. Now, this looks like wonderful. Hey, all the orange is on the right and all the blue is on the left. It's important to kind of temper our expectations at this point, though. This is a discovery set, and we've trained our classifiers very strongly on this discovery set, which is a small set of samples. It's overfit. It's something you hear a lot, but what it means is that we've managed to classify very, very well in our particular sample, probably by honing in on things that are not relevant. So some of this classification is too good to be true. And what we'd like to do is sort of think about what is a more realistic expectation, I guess, for what our classification performance can be. And that's where this validation aspect comes in. One thing that is one approach that we can use is to simulate different, what I call population plausible data sets using sort of features in our own data. One of the ways we do that is we fill in missing data using multiple imputation. We do simulation by resampling and so forth. And kind of a very simple example is a leave one out resampling, where you just leave one observation out and you try to use all the other observations to classify that one. And notice that you can get some kind of a guess from that. We have a bunch of informed guesses here, like we plot different points of sensitivity and specificity for each one of these sort of simulated data sets and take the average of those, run a curve through it, and then get a guess for what our classification performance will be, in this case, the AUC. So rather than the really high AUCs that we observe in our sample, this AUC is sort of a predictive value, a projected AUC, if you will. It's a guess about what the future performance will be. In an independent sample, that's none of these. We're also, in order to rank the candidates, we want to kind of try all different groupings of them, but we don't know which groupings are the right ones. So we use another technique called Bayesian model averaging, where we run a large number of different models and then average those results from those models. This means that we don't have to really specify the correct model. We just have to know that something in there is relevant. And we also look, in this case, we want to compare to if there are any existing biomarkers. In this case, there's a genotype marker, ApoE genotype, that is somewhat predictive of Alzheimer's disease risk. So do our markers add, make an independent contribution to that? Like, for example, if we were to know, if we already knew the ApoE status of a patient, could we also add the information from our markers, and would that improve our prediction? In this case, you can see that it seems like maybe it will. Maybe it will by a lot. And finally, so now we've got kind of a smaller set of markers. In our case, we ended up with a couple of dozen, three dozen sort of top candidates. And we drew now an independent set of samples. And we want to see if our markers kind of hold up in the independent set. But important thing to do before we even jump into that is to look at the shape of the response distributions. That, like I said, we're dealing with small samples here, and the samples may not be as comparable as we think they are. Just because we're sampling from cases and sampling from controls doesn't mean we've got a fair sample in each case. There may be, just sampling-based differences in the two samples that are unrelated to the problem at hand. In others, the differences in the samples are not really related to the association of these markers with the disease, but those features may be influential on the markers nonetheless. So again, this is getting at the issue of robustness. If we find issues like that, it's an indication that the markers are less robust than we thought, and we might want to downweight those or deprioritize those. Some of the things that we can look for are features of the data that are called skew and kurtosis. Skew is kind of roughly whether the distribution kind of is heavy in one side or the other instead of being symmetrical. And kurtosis is kind of like how pointy or centralized a distribution is. If you've got really high kurtosis, most of the values are very close to one another in the middle, with possibly some large outliers. Low kurtosis means that it's kind of more diffuse and spread out, less centralization. So here we've got an example of, in our particular case, I'm just plotting here 26 markers, which I've just labeled A through Z, and where we sort of have a distribution of their CT values and kind of compare what we found in discovery to what we're finding in this validation set. And for the most part, they sort of line up, but there are a few cases where they don't. In some cases, all of the values in the old cohort, in other words, the discovery cohort, fell on the extreme edges of what we're seeing in the new cohort. That indicates that maybe the old cohort, the discovery group, had some outliers. There could have been some large skew. Also, for some of the markers, we're seeing sort of kind of thinness in the middle, like there's kind of more on the edges than there are in the middle. That's an indication of low kurtosis, or another way to view that is that maybe we've got sort of dissimilar groups mixed together. So our cases could be two kinds of cases, or our controls could be two kinds of controls. In other words, some controls that are not going to develop the disease and other controls that maybe will. Those are things to look for and worry about. Another important thing before sort of concluding that, hey, we've got valid markers, is to see if our basic assumptions, like our relevance assumptions, are still true. For example, some of the markers we selected to be non-discriminating, and those are, we were calling sort of positive controls here. That means we don't think they should distinguish the groups. So are they still non-discriminating in the valid set? Were we correct in that assessment? So one way we can do that is this is taking what's called a Mahalanobis distance. It's a technique for sort of combining all of the markers into a single space and looking at distances. Notice the prediction value, the AUC, I guess, which is roughly the probability that an AD value will be further away from the middle than a control value would be. is the probability is really close to 50%. That means they don't discriminate. So the ones that we don't think should be discriminating turn out not to be. And notice the blue and the orange are all mixed up in the middle. That's what we'd like to see for this kind of set of markers. Now for the ones that we chose to be discriminating, the ones that we're sort of targeting, these are our actual candidates. Are they still discriminating? And here notice that the probability that kind of an orange one will have a large distance from the middle is much, much higher. So notice that most of the blue tend to cluster toward the bottom in sort of the typical middle range. And all the ones that are atypical, or at least most of the ones that are atypical, are orange. So that's indicating that, yeah, these markers are giving some information about the disease status. So they remain relevant even in the new independent sample. Also, we may have differences in how the classification plays out in the discovery set and the validation set. In fact, it's quite common to see that. So we want to see if the patterns, or in other words, the shapes of the rock curves are similar. Based on our discovery data, we could make some projections, some guesses. So the green lines were all of sort of averaging our microRNA models. This is like what I call the Bayesian model averaging. This is the guess that we got for what our performance would be in a new cohort. It should have this particular shape. So we think that the curve, the green curve, will sort of pass through that little box, basically. So does it pass through that region? And when we look in the results, we see that it does. So the microRNAs, at least as a whole, are performing pretty much as we expected them to perform. So we continue to have a relevant set, and this is kind of confirmatory behavior. Another thing that we looked at is, well, wait, what if we add in the genetic marker? Where do we get the increase in diagnostic performance, diagnostic power? Our indications in the discovery phase were that it actually wasn't all that helpful, that most of the diagnostic bump occurred at sort of low specificity ranges. So look, we get kind of a large separation in those. The purple is much higher than the green, but only when like specificity is like 50% or more. So that's not all that helpful. We'd like to choose a marker that has higher specificity. And our initial results suggested that maybe the genetic marker would not be all that useful in the eventual panel. But when we look at our validation set, that pattern flipped quite a bit. Now we're getting all of the bump in very, very high specificity ranges, and that's an interesting and helpful finding. What could explain that? Well, we haven't fully tested this, but one idea is that our discovery cohort was less healthy. In other words, the cases were more severe cases. That actually is true. When we went back and looked at both cohorts, we found that the discovery cohort had a lot more examples of extreme disease than the validation cohort does. So in other words, when your symptoms are already very prominent, the genetic information, which predicts your future risk, is less important. It doesn't add much new. But when you've got a severe case of disease, for example, maybe a latent case, or it's just starting to kind of set on, then the genetic information can take on more predictive value. So it's important to look at this kind of nuance because it'll help us decide not only who we should apply the biomarker screening to, but also when. Like at what point in the disease is it going to be valuable to consider genetic information or not, consider the markers or not. Finally, so once we've kind of looked at all this in a global sense, now we'd like to continue to pursue these markers, but we want to prioritize them. We want to think about which ones are really the most promising, the best ones to move forward with. And so, again, we're going to look at that in kind of a consensus type way, where we use our same methods as before, evaluating all the markers, but before we use those the consensus methods to exclude things, right? We wanted to just sort of throw away the ones that don't work and keep the ones that do. Now we're using them to rank and prioritize. And so it's important to note, too, that one of the judges one of the rankers is our discovery data. So we include that as also a judge. So our discovery data gave us a prioritization. That was what we decided at the very end of the discovery of the UH2 phase. So how well do those rankings hold up? but those are also important because that was a different cohort, and it represents some information that the new cohort does not have. So it's important to incorporate that as well. The way we organize all this table is basically each judge, for example, each method is, we just tell it to rank all the markers. You rank it according to whatever that method does. In this case, for the CART random forest, it would rank it according to how well or how important that marker was in prediction models. So each marker is rated best to worst, and then for each marker, so for, in this example, marker I, we would sum all of their rankings across all the judges to get a value here, 70, that would be the sort of, that's their value, I guess. That's what we call the rank sum. You can also notice that we've also color-coded the table so that we can see how consistent the rankings are. In some cases, like look for, you know, markers A and B, the rankings are extremely consistent. Also for marker Z, most of the methods don't feel that that's an important marker, whereas A and B really are. Once we have those rank sums, that forms kind of an ordering, which is now our consensus opinion. Across all these evaluation methods, this is what we believe the priority of the markers are. I've obviously labeled them A to Z according to their priority. But notice here that we can also sort of group them, like, you know, into maybe blocks of high priority versus low priority markers. We don't have to necessarily select them one at a time, but we can think about which groups of markers to prioritize. And I'm going to kind of skip this in the interest of time. It's kind of a technical detail. Basically, what we've got is kind of a block of what I call hot markers, the ones that seem promising, some lukewarm markers, ones we're less sure about, and cool markers, ones that are not as promising. So how well does this sort of classification hold up in terms of when we start to combine? This is, again, these are kind of evaluations of the markers as important in predicting. What if we try and combine the markers? Do we find that the marker prioritization is relevant to predicting? So in other words, if they're as important as we really think they are, they should contribute the most to models. They should be the most important variables in selection models. So here we have got basically a test of that. We try all possible parsimonious models, like models that are not too large, and we color code them by how many markers are in the model. The best ones are the ones here in the upper left corner. Those are the ones with the highest AUC and the lowest AIC. AIC is kind of a measure of how well the model fits, how parsimonious it is. So which markers are in that best model set? Which ones are contributing the most? And it turns out that are the highest ranked ones, in particular the top half. The ones ranked from, say, like 1 through 12 or so are the ones that are contributing the most. Those going back here are pretty much our hot markers and a few of our lukewarm markers. So this is kind of an internal validation that our prioritization is holding up. And it also holds up in sort of a larger sense of like discriminating our groups, classifying the groups. Remember, I said in this validation cohort, the cases were less severe cases. So the problem is actually harder in this data set than it was in the discovery data set. And we also have sort of weaker classification performance, but there's some indication that although maybe some of those cases, maybe we have some cases in the population that our markers are never really going to work for, there are probably also situations where the controls are just latent cases. They just haven't expressed yet, and our markers are possibly predicting something valuable about those. So by, for example, tweaking the cutoffs, we may be able to capture some of those. Another thing that we want to look at in order to sort of help prioritize moving forward is to get a sense in a population setting, how good could our best possible classifier using these markers be? And this is kind of where we're thinking about, for a variety of cutoffs, what can we expect in terms of sensitivity and specificity? And we've got sort of a range that we're thinking about. Notice that if we don't have enough markers, we can't really do any better than, say, the genetic marker already, the ApoE status already does. We've got to add several more in order to reach kind of to get to a point of clear discrimination. In other words, the markers need to be chosen so that they complement and enhance one another. Lastly, we're also, like I said, looking for covariate effects. And here is just kind of simple things. We haven't tested this extensively, but one way to kind of look for clinically relevant covariate subgroups, like groups where the markers may perform differently, is just to sort of cluster the subjects in some way. Notice that I've clustered it 3 ways here. The cluster 3 is almost 100% AD, so that's a very sort of strong AD subgroup. And the rest of the ADs are scattered amongst clusters 1 and 2. But notice something interesting. Cluster 2, the AUC, that's the classification performance, is higher. It's about 0.78. That's pretty close to what it is in cluster 3, versus in cluster 1, it's only 0.56. So something is different about the cluster 2 patients that makes the markers more sensitive. And in cluster 1, the markers are not as sensitive. One of the differences is that there's more males in cluster 2. So there could be a possible sex interaction going on. Again, that's just a guess. We're hypothesizing. It's something to test in a future study. But this kind of thinking, looking at this more exhaustively over a large number of covariates, can also give us more predictions or more hypotheses to test. Also, we want to see if, and here's back to the question of when we should apply these markers. If we've got some kind of like spectra, like a spectrum in a covariate, for example, in the case of AD, one of the things is the mini-mental exam, where this is some kind of like a diagnostic tool that says how bad their cognitive performance is. And as the cognitive performance worsens, it becomes more and more likely that they have Alzheimer's. So are our markers trending in the right way? Do they start to signal better for more severe cases? But the problem of looking at this for any of these covariates is that the relationships could depend very much on the marker. It may not be our whole marker set that's sensitive, but just one of them. Or they could be nonlinear in some way. So in this kind of simple example, let's say this is one of our covariates, I won't say which, but the higher marker ranks do seem to correlate with kind of higher association of the covariate to the marker itself, so that we see the darker red on the right and kind of closer, moving toward orange and yellow on the left. But that's only true for the top half. So that in other words, only in the strongest markers. In the weaker markers, we don't see a pattern like that. It's much more blotchy, so it's less clear. Anyway, that is essentially my talk. An abbreviated part of this, in other words, just the discovery part of this pipeline, was presented in a paper that was published earlier in the year in Journal of Alzheimer's Disease. There's a reference to that right here. We're currently working on a paper on the validation aspects of it, and then also a standalone methods paper that essentially is the content of this talk. So that's all. Thanks for listening. Thanks, everyone. And if you have any questions, I'm happy to open up to questions right now.
Jack, this is Matt. One question. You mentioned at the start the methods you went through versus, say, false discovery rate or other methods. Has there been any comparison to compare those methods?
Actually, no, although that's something that nothing that I'm ready to present. Yes and no. So yes, it's something that we're going to address in our methods paper that I mentioned at the end of the talk. But no, it's something that we've gotten to examining in a lot of detail yet. It's sort of one of the things that's still on the agenda. However, I should say that we have done a comparison in a different setting, not this problem, another problem that also was involved in a selection methodology similar to this one, and did that comparison and found that, for example, the usual false discovery rate corrections were highly optimistic. So our methodology gives a much more realistic and pessimistic view of what can be expected in an independent data set. So a lot of I would say that our methods are more conservative in that sense. But in this particular application, I don't have any formal comparisons yet. Yeah.
Thanks.
Jack, could I ask you a quick question about study design and influencing the way that you would apply some of these statistical methods?
Sure.
So I guess in terms of if you have a matched case control, and it's not simply matched on age and sex, but now you start to match on additional parameters, where some of the traditional teaching is that you have to look at it with specifically sort of a conditional logistic regression. How would you be able to apply some of these other test parameters, whether it's random forest or looking at the ROC curves, when you've matched on several factors that actually are known predictors of outcome?
Yeah, that's a good question. One kind of way that you could go about it is to just simply not worry about the fact that you've matched on many other things, and then interpret all your results as conditional on that matching. In other words, assuming that you've got, so your association values or something like that would be conditional on having done that matching already. And so you can interpret it in a conditional sense. In a diagnostic, like screening for disease type of setting, what that would mean is that you would first learn some things about all your patients, like you'd know how old they were, you'd learn their sex, you'd learn some blood panel values or whatever, and you'd place them in a bin beforehand. And then depending on what bin they were in, you would run possibly a different diagnostic test, if that makes sense. So that's the more subtle way to kind of go about it as well, is if you have some prior information where you really know that, for example, prediction using your set of markers is much more sensitive within some groups than it is in other groups, then kind of a usual approach is to develop diagnostic screening criteria differently in each of those groups. So you could sort of take each group as kind of its own set and develop sort of thresholds and cutoffs, or possibly even choose different markers depending on the specific features of that set. It's a much more complicated problem, though, because it requires, well, one, it's more expensive. It requires larger sample sizes, especially if you're matching on many different things. But also, it requires some fairly sophisticated knowledge about how your markers are sensitive to those covariates that you've been matching on. Usually, that kind of information is not available early on in a discovery setting. So in that sense, I really wouldn't recommend doing that in advance unless you have really, really good knowledge that that's true. If you do have really good knowledge that's true, then possibly you shouldn't be looking in such high dimensions anyway. So this kind of discovery type methodology may be less relevant in those cases because you've already done some sort of selection and tuning, I guess, of your problem to begin with. But yeah, I mean, to give just a simple answer to your question, I think if you've got really, really strong matching on a large number of covariates, my first inclination would be to look at the screening selection independently within each subgroup that are defined by those strata.
So Jack, I have one quick question. So have you seen any of the miRNAs that would be regulating the gene expression of ApoE or beta amyloid precursor protein?
Right. We have been looking specifically for that, and nothing as compelling as you might think. So one sort of really interesting finding is that all of the microRNAs that are among our top, most highly prioritized candidates all seem to be targeting the same protein in some sense. And so that's very kind of compelling to us, but as far as we know, that's not related to amyloid plaques or other things. So we're still sort of exploring that.